I am Dr. Moumita Rai, Assistant Professor, Department of Pharmaceutical Technology, JISU University. The topic of today's lecture is genome organization. This is the eighth topic of first module in the cellular and molecular pharmacology subject code MPL104T. In today's class, we will be discussing about genome, what is genome, and organization of different genes. So first of all, we have to know what is genome. Genome is basically the genetic material of any organism which mainly consists of coding as well as the non-coding DNAs. The coding DNA are mainly responsible for the synthesis of protein, and the non-coding DNAs are acting mostly as the linker DNA, which we will be discussing later on. And the entire set of genes which are present in the haploid state is called the genome. But in case of the diploid organism, they consist of two set of genes, that is two genomes. And the haploid human genome is containing approximately 3 billion base pairs of the DNA, which are packaged into the total 23 chromosomes in it. So now the genome organization. What is basically the organization of the genes or the genome organization in any organism? The DNA is basically the negative charged, which is due to its phosphate group. Due to its phosphate group, it is negatively charged. And these DNA are packaged by the histone protein that is comprising this chromatin structure. In the nucleus, the chromatin structure is mainly consist of the DNA which is packaged by the histone proteins, where the DNA is negatively charged due to the phosphate group, and the positively charged histones are making the DNAs in a packaged format. These histone proteins are mainly of 5 types like H1, H2A, H2B, H3 and H4. These are mainly 5 types of histone proteins which are helping with packaging the DNA. Now there are different types or different forms of these packaging system. Like in the picture we can see there are three terminologies. One is the linker DNA, another one is the nucleosome, another one is the chromatosome. So the linker DNA is basically the H1 histone protein plus 20 base pair of DNA. DNA plus H1 histone protein, this is making this linker DNA. And the protein octamer means the rest 4 histone proteins like H2A, H2B, H3 and H4, each in 2 set, means H2A into 2, 2 H2A and 2 H2B. Similarly, 2 H3 and 2 H4, these total histone protein means 4 into 2. These 8 histone proteins are making this protein octamer. Octamer means 8. So, these 8 histone proteins, 2 of each, these 4 histone proteins are making this protein octamer. Along with that, 146 base pair DNA. These totally making this nucleosome. This is one total nucleosome which is basically consisting of these protein octamer like this histone protein structure and the DNA. And these linker DNA with this nucleosome. Like in the picture in this part you can see the linker DNA with this nucleosome. These all together is called the chromatosome. Okay, so the chromatosome is the nucleosome plus linker DNA. So in case of linker DNA, we have just learned it is containing H1 histone protein and 20 base pair DNA. And in case of these nucleosome, it consists of these rest of the 8 H1 proteins, rest 8 histone proteins and this 146 base pair DNA. So this linker DNA plus nucleosome means there 8 histone proteins are there and 1 histone proteins. So total 9 histone proteins and 146 base pair DNA and 20 base pair DNA. So near about 166 or 164 base pair DNA. This is totally making this chromatosome. So linker DNA is the DNA plus the histone protein H1 protein, and the rest of the 4 histone proteins in 2 of each, the protein octamer plus 146 base pair DNA is making this nucleosome, and nucleosome plus H1 histone protein, total these linker DNA is consisting of this chromatosome. So this is basically the genome organization. Now coming to another term, which is the repetitious DNA. So what do you mean by the repetitious DNA? That is basically the DNA which is just in repetitive format. Okay, so as the DNA is a double helical structure, so they are always associated with each other. The two strands of the DNA are associated with each other. If there is separation between these two strands, that is called the denaturation. Denaturation is the separation of two DNA strands. And again, reassociation and complex formation is called the reassociate. Renaturation and complex formation is called the reassociate. And this rate of reassociation is totally depending on the DNA repeat copy. When and how much rate of these reassociation will be, that is depending on the DNA repetitive copy, or the DNA repetition is totally depending on this rate of reassociation, in which rate the two strand of DNA will get reassociated, that is depending on the DNA which will be making its repetitive copies. So on the basis of that, the type of DNA can be classified into mainly three types of DNAs on the basis of their reassociation rate, and there are some characteristics of these type of DNA which we will now discuss. First is, whenever there is a slow reassociation rate, like the reassociation or the reconstitution rate or the double helical structure formation rate is slower, then there will not be any repetitious DNA. As the rate is very slower, this is totally directly proportional, like the rate is very slower, so there will be the single copy of DNA which is non-repetitious, and about 5% of the DNA will codes for the proteins, and the remaining has not such a known function, which is referred as the spacer DNA. Spacer DNA and protein coding DNAs are there. Mainly 5% are the protein coding DNAs, and they are the non-repetitious. These mainly comprises of 50 to 60% of DNA are of this type. Then the second one, which is the 25 to 40% of the DNA, that is the moderately repetitious DNA. Means their repetitive rate is intermediate degree and they are called moderately repetitious. So from the term we can assume that the reassociation rate is also intermediate. They are having the intermediate reassociation rate, so they are moderately repetitive. And the last one is the rapid reassociation rate, having the DNA which are mainly 10 to 15%, which are having the rapid reassociation rate. So they will be the highly repetitive DNA and termed as the simple sequence DNA. So the highly repetitive DNA are called the simple sequence DNA. The DNA which are having the intermediate reassociation rate are called the moderately repetitious DNA, and the slow reassociation rate having DNA which are non-repetitious are called the spacer DNA, and plus this non-repetitious DNA are 5% of that are protein coding DNAs. So the simple sequence DNA or the highly repetitious DNA are mainly the long stretch simple sequence DNA, which is called the satellite DNA, which is used in case of the DNA fingerprinting. As we know that DNA fingerprinting is an important tool for these forensic department for the identification of a particular person from their fingerprints, and all fingerprints or any type of body parts, body fluids or anything, crime suspension can be checked or for with this help of this DNA fingerprinting technology. And another thing is the confirmation of the parenthood. The identification of the father and mother can also be done with this DNA fingerprinting, and that is the usefulness of this simple sequence DNA, which is in higher organism 5 to 10 base pair long repetitive sequence, which may long up to 10 to the power 5 base pair. That is one long stretch simple sequence DNA, which is mainly called the satellite DNA, and every individual are having unique in number and length of this repeat unit of DNA. So that is one important characteristics of this DNA from which the identification can be done, that can be distinguished between one person to another person with these simple sequence DNA basis, as every individual is having the unique number and length of this repetitive unit. Now the next one is the repetitious DNA, which is in moderate form, that is the moderate or intermediate repeat DNA, which may be divided into two types mainly. One is the lines, another one is the signs. Lines are the long interspersed elements and the signs are the short interspersed elements. These interspersed elements are basically the transposons, or called the jumping DNA, or the jumping sequence basically, which can translocate from one position to another position. And these lines are having the repeat unit up to 7,000 base pair, which can go for translation transcription to form protein, and they are mostly the transposon, which we will be discussing in the later on slides. And the signs are the short interdispersed elements, which are containing the repeat unit of 500 to 700 base pair, and they are mainly the non protein coding, mostly the retrotransposons. Now the organization of genes, if we are coming to discussion of these genomic organization, the simple sequence DNAs are mainly comprising these solitary genes, which are repetitive in only once, and the 25 to 50% of protein coding genes are there. Like here is one sequence, then after 20 kb there will be another repetition of the sequence. There will be one repetition, and that is the simple sequence DNA, which is comprising these solitary genes. Hmm? And these protein coding genes are only once it's repeated in the haploid genome. Next is the duplicated genes. Duplicated genes, or the gene family and the pseudogenes, these terminology we will be discussing. Here about 50% of the protein coding genes are made of these moderately repetitive DNA, that is the duplicated genes, and DNA with 5 to 10 kb of a gene has a sequence which is similar but not a directly copy of that gene. Means it is similar gene or sequence. Duplication of the ancestral genes means carrying out the hereditary information but non-identical. They are similar but not exactly identical. The similar genes or the sequence are arises by the duplication. They are very similar but non-identical. A set of these duplicated genes which encodes protein with similar but non-identical amino acids are called the gene family. A set of these type of duplicated genes which are similar like the ancestral genes and encoding the protein, but they are non-identical amino acids containing, they are called the gene family. And the duplicated genes which are non-coding protein type of genes, that are called the pseudogenes. Means they are the duplicative genes but non-protein coding. They are the pseudogenes. And another term is the tandemly repeated genes or tandemly repeated genes. They are having the multiple copies of gene in cell codes for the rRNA, tRNA and histone proteins. They encodes for the identical protein or the functional RNAs. In just contrast to the previous genes, that is the duplicated genes, where they are similar but non-identical, and these tandemly repeated genes will encode for the identical protein, and they are very important in active protein synthesis. They are present in multiple copies in back to end over a long stretch of DNA, and these are very important in cellular demand during these active protein synthesis. So from these slides you have to know the terminologies like what are the solitary genes, duplicative genes, tandemly repeated genes, then the simple sequence DNA, lines, signs, these terminologies you have to remember. Next is what is the transposons or the transposable elements or the mobile units. The DNA sequence is changing its position within a genome and altered the genetic identity or genomic size, that is called the transposable elements. The DNA sequence which is changing its position within a genome, which leads to the alteration of the genetic identity and genome size, it's called the transposable elements. They are capable of moving one position to another position, that is the jumping gene or mobile element, and non producing any physiological functions, maintaining the identity only. They are called the selfish DNA, as they are present within the genome and maintaining only their identity. They are not beneficial or not producing any type of physiological function, but they are present within the genome maintaining only the identity. So they are called the selfish DNA. And DNA are transferred from donor to the target by directly as the DNA transposons or indirectly by the RNA retrotransposons. Means transposons are the donor DNA plus the transposase which is cut to release the DNA segment, and the DNA segment is finally binding with the target DNA, which we will be discussing in our later slide. In the picture, just in the picture you can see, like in this, the DNA transposons where the is donor DNA is just binding with the transposase and excision is done. By these transposase enzyme, this excision is done where, where there is a cut to release the DNA segment. The DNA intermediate segment is cut and this segment is binding with the target DNA. From this donor DNA, one segment is excised and that is binding with the target DNA, and finally that is forming the transposable elements which is within this target DNA. But in case of the retrotransposon, from the donor DNA, first it is transcribed to the RNA. RNA intermediate, it is transcribed. Transcription is done, it is forming the RNA intermediate. Then again with the help of the reverse transcription, it is forming the DNA intermediate. Then it is integrated to the target DNA and the transposable element is that. So that in case of the DNA transposon, from the donor DNA, direct the segment is excised and it is binding with the target DNA. But in case of this retrotransposon, from the donor DNA, it is first transcribed to the RNA intermediate. Then from it again the reverse transcription process is done, and then the DNA intermediate will be binding with this target DNA. That is the total process of this transposon. So till now we have discussed about the organization of gene and what is the genome and what are the different mobile unit or the transposons, and in the next class we will be discussing about the gene expression. What is the gene expression system or the what are the different steps of gene expression. So till then just remember these questions. One is what is genome? Next, write the organization of genomes. Like in the organization of genomes, we have learned the different terminologies and also the simple sequence DNA, moderate or intermediate type of DNA, these type of DNAs. Next, the repetitious DNA, which are consisting of these simple sequence and moderate DNA. And genome organization is mainly the histone protein containing, and with these the protein octamer is forming, and with the DNA the nucleosome chromatosomes are forming. So these type of things you have to know very well, so that in the next classes you will understand the detailed process. Next is write about the organization of genes like the solitary genes, duplicated genes, pseudogenes, satellite DNA. These type of terminologies you have to also remember. And the last one is describe the transposon with its type. So this is all about today's class. Read this very carefully. If there is any doubt, then please ask me in the next class, and in the next class we will be continuing with the gene expression. So till then, thank you so much. Thank you for joining.